Hi, everybody. Welcome to this last set of video lectures, and it's going to be a set of two videos. This is the first one, and it's going to be on DNA structure, and it'll be followed by another video on DNA replication. So let's get started. Okay, so we've been talking about DNA in sort of a bigger bird's eye view kind of way. We've talked about moving chromosomes around in the context of mitosis. Chromosomes are made of DNA, right? We've talked about replicating DNA in the cell cycle. But here in this video, we're going to zoom in on the actual molecular structure of the DNA itself. What does it look like on a molecular level? The name DNA, it stands for deoxyribonucleic acid. So the D and the N and the A altogether, DNA is the shorthand. And DNA is a member of a family of macromolecules called nucleic acids. And this is actually the last family of macromolecules that we have to talk about this year. You might remember that we've talked about fats in the context of phospholipids in the cell membrane. We've talked about sugars and carbs in the context of cellular respiration. And we've talked about proteins. And the last macromolecule family is nucleic acids. And DNA is a member of that family. So in those other macromolecule families, especially when you think about, for example, proteins that we just learned about, we talked about proteins being a sort of polymer, right? A large molecule that's made up of subunits. And DNA is the same way. It's made up of subunits, and it's the large molecule, and it's got individual pieces that make it up. And when we were talking about proteins, the subunits were called amino acids. For DNA, our subunits are called nucleotides instead. So nucleotides, there's a picture of one right here. You can see that it has three different parts. So there are three components to one nucleotide. And let's go through those three different parts. One part is a sugar molecule. So you might remember that when we were learning about monosaccharides, those simple sugars, they all had that ring shape. And you can see that this sugar, the central part of a nucleotide, has that same ring structure. And then connected to the sugar on either side, there are two different things. One side has this molecule, a phosphate molecule, and we've run into phosphates a few times before. We saw phosphates in the phospholipid head, and we also saw phosphate in ATP, adenosine triphosphate. So we've run into phosphate a couple times before, and it's coming up here again on one side of a nucleotide. And then on the other side, we have this component called a nitrogenous base. And the name is kind of a lot, but really all it means is that it's a structure that's got a lot of nitrogen atoms in it. So nitrogen, nitrogenous base. And this nitrogenous base is actually really important. It's a really important component of a nucleotide because it is going to be where all the variation for nucleotides will occur. This is sort of similar to, if you recall, the structure of, let's take it back to proteins. Proteins are made up of amino acids, and we said there are 20 different kinds, right? And that's what gives proteins their variety. And in each amino acid, it's different because of that R group that was hanging off of the side. For nucleotides, the part where the variation is going to occur is going to be this nitrogenous base. And we don't have 20 different kinds, we only have 4, but it does still give us some variety. So let's take a look at what those 4 are. So here's a picture of our 4 different nitrogenous bases. So we've got thymine, we've got cytosine, we've got adenine, and we've got guanine. So just remember that for each of these, thymine, adenine, cytosine, guanine, we're only changing out this section of the nucleotide. So only the nitrogenous base is changing. Okay, so as you look at these pictures, you might notice a couple differences between these molecules. And the most obvious difference is that the ones on the left are made up of a single ring, whereas the ones on the right are made up of this double ring. And we have words for these two different types of nitrogenous bases. The kind that are made up of only one ring are called pyrimidines, and those include thymine and cytosine. And for nitrogenous bases and nucleotides, we almost always refer to them with the letter. So thymine will be referred to as a T, and cytosine will be referred to as a C. And then the larger ring structures that are made of two pieces, I guess, two rings, are called purines instead. And those are adenine, or A, and guanine, or G. So these are 4 bases that that we have as our options to slide into a nucleotide. Okay, so let's take a look at how each individual nucleotide is going to connect to the next. When I'm going to keep making references to proteins. For proteins, they're made up of strings of amino acids that are all connected to one another, right? So in the same way, our DNA is going to be made up of strings of nucleotides. But the nucleotides are going to be connected to one another in a very specific way. So if you take a look at the, I guess first let's look over here on the left, you can see our diagram of a nucleotide. We've got a base, we've got a sugar here in the middle in purple, we've got a phosphate over here to the left, and then we've got a base over here on the other side, and we've blown up this base section to show the 4 different options that we have at this position. Okay, so this is one nucleotide. And if you look at this diagram to the right, you can see that what I'm circling right here is also one nucleotide. It's got the same sugar, this purple pentagram. It's got the phosphate to the one side, and it's got the base to the other side. And this is showing, it looks like adenine is connected over here. So this is one nucleotide. And right below it, we have another nucleotide. Again, we've got the sugar, we've got a phosphate to the side, and we've got a different base. This one looks like cytosine. So where they're connected, you can see, hopefully, that the sugar of one nucleotide is connected to the phosphate of the next nucleotide. And that nucleotide sugar is connected to the phosphate of the next one. So we get this sort of phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar strand. And people refer to this as the sugar phosphate backbone of DNA. And part of the reason it's called the backbone is because the bonds that hold these molecules together are they're quite strong. They're called covalent bonds. So we get this sort of straight line of sugars and phosphates connected to one another. But remember, sugars and phosphates aren't the only thing that a nucleotide is composed of. We also have the base. And so these bases are just kind of sticking out to the side of each sugar. So you can think of the sugar molecules as being sort of like the central component of each nucleotide. And each sugar for a nucleotide is going to connect to the phosphate of the next nucleotide. And then that nucleotide is going to have its own base, and so on and so forth. And then the one last point, we're showing here a stretch of a little bit of DNA that's 4 nucleotides long. We've got 1, 2, 3, and then 4. But actual DNA molecules tend to be hundreds and more often millions of nucleotides long. So it would go on much longer than this page. We're just showing sort of a zoomed in short little piece here. Okay. There's one last slide about DNA structure itself, but it's a doozy. So let's look at this one more time. So you'll notice that we have one strand here. So there's one sugar phosphate backbone. Each nucleotide is connected to one base. Each sugar is connected to one base. So we have one strand of DNA with one backbone. But actual DNA is actually composed of 2 strands of nucleotides. So I think it's, you can see it in all of these diagrams. This blue ribbon structure looking thing is going to be the backbone. And you can see there's one strand, and then next to it, another strand. So there's 2 strands. And in this diagram here, these light blue rectangles are the sugar phosphate backbones. And so there's one strand here, and there's another strand over here. So DNA, as it occurs in our cells, as it occurs in nature, is not a single-stranded molecule, it's a double-stranded molecule. And one helpful way to think about the structure of sort of what it looks like is to think about a ladder. So it kind of looks like a ladder in this diagram right here, right? So the sugar phosphate backbone is going to be sort of the side of the ladder where you would put your hands. And then the rungs of the ladder where you'd put your feet are going to be composed of the two bases next to one another. So each nucleotide, each DNA strand is still going to have its sugar phosphate backbone and its nucleotides sticking out to the side. But the outside is going to be where the sugar phosphate backbone are. And the inside is going to be where that those nitrogenous bases, where they're acting as the rung of the ladder. And then finally, it's not just a straight ladder like up and down. It actually has a twisted shape. So if you took this ladder and just gave it a twist, that's what the DNA actually looks like, is this twisted ladder shape. And the word we use for that is a double helix. Okay. So the next thing I want to discuss is what are called pairing rules. And this refers to the fact that in DNA, you always find specific nitrogenous bases next to one another. So we said that the nitrogenous bases are immediately adjacent to one another. They're right next to each other. And you're always going to find the same bases sort of paired up. So A's are always going to go with T's, and G's are always going to go with C's. So if you look at this strand right here, anytime you see an A, there's going to be a T next to it. So here's an A, there's a T next to it. Here's an A, there's a T next to it. And anytime you see a G, you'll see a C next to it. So here's a G, we have a C. Here's a C, we have a G. They always go together. And it doesn't matter which side of the ladder they're on, right? So here we have a G on the left and a C on the right. And here we have a C on the left and a G on the right. But wherever you find one, you're going to find its pair, its match. And this doesn't say anything about the order that the nucleotides go in along one strand, right? So this could be an A, a T, a C, or a G. This next one could be an A, a T, a C, or a G. But whatever one you find, you're always going to find the match on the other side. And the word we use for this is, we say that these bases are complementary to one another. So G is complementary to C and A is complementary to T. And when you think about this word complementary, I think when you think about like food, like complementary foods, or like a drink that complements a certain dish, it doesn't mean that they're the same, right? It just means that they pair well together. They go well with one another. And in the same way, A's and T's go well with one another, and G's and C's go well with one another. They pair up. So we can call the bases complementary, and then we can also actually call the strands themselves complementary. So since all of the bases on this left strand are going to be complementary to all the bases on this right strand, we can call the strands themselves complementary to one another. So both the bases and the strands are complementary. Okay, two more points to make about this. So one point is that I talked earlier about, for the sugar phosphate backbone, we have those strong covalent bonds that are holding it together. But the actual, when you look at what's holding the two strands together, so we have a strand on the left and a strand on the right, and what's holding those two strands together, holding them to one another, is not a strong covalent bond. It's actually a very weak, it's called a hydrogen bond. And the name hydrogen bond is a little bit of a misnomer. It's not actually a bond. It's just an attraction. But if there's an attraction between all of the bases on the left and all of the bases on the right, then these two molecules are going to stick to one another, right? They're going to be attracted and they're going to be held together. But those bonds themselves are very weak. So if you think about all of the solid lines over here are representing covalent bonds, which are really strong, sort of like superglue bonds. And if these covalent bonds are superglue, then these hydrogen bonds are sort of like an old glue stick that's been left out for too long and it doesn't really work very well anymore, but it's still a little bit sticky. So it gets the job done. The two strands are held together with these weak bonds. And then lastly, there's this word that's getting blocked up as soon as I went over to it, but the word is anti-parallel. And so this word, if you break it down, one piece of it is parallel. So in the same way that two lines can be parallel to one another, the DNA strands are parallel to one another, right? They go along the same track. But DNA strands have a polarity to them. So if you see, I think it's most obvious on the sugar molecule. The sugar molecule has an oxygen on one end. And on this piece, the left side, the oxygens are all pointing up. So we have oxygens on the top and a phosphate on the top as well. But if you look at this other strand on the right, these oxygens are instead now pointing downwards. So the oxygens are pointing down and the phosphates on the bottom. So they're oriented parallel to one another, but in opposite directions. They run in opposite directions relative to each other. So the word for that is anti-parallel. Okay, we've got one more slide for this video lecture. And that's just, I want to make the point that these DNA, we have so much DNA molecules in our cells. It could go to the sun and back. I think it's like 600 times or something like that for one cell. So when we're thinking about all of this DNA, it has to be packaged up into, at the very least, chromatin. And then if it's getting condensed even further, it's going to become a chromosome and take on this familiar shape. And the point here is that one chromosome, so an unreplicated, this is showing a replicated chromosome, but an unreplicated chromosome is a single piece of DNA that's just really, really, really long. So when you're looking at a single unreplicated chromosome, that is a single piece of DNA. And then once the DNA, or once the chromosome replicates, or once the DNA replicates, then it's going to become two pieces of DNA instead. And we're going to explore this idea of DNA replication in the following video. So I will talk to you guys soon.